Okay, today we're going to talk about organic molecules. What separates organic chemistry from inorganic chemistry? It depends on the actual compound that you're studying. The organic molecules, the organic compounds, they always include carbon joined either to itself or to hydrogen. So you've got to have both carbon and hydrogen for an organic molecule. Now, of course, we're in biology. We're interested in talking about the organic molecules that are in living things. And we refer to these as biomolecules. So these are organic molecules. They all contain carbon and hydrogen. And there are only 4 biomolecules. There's lots of different types of these biomolecules, but only 4 separate different ones: carbohydrates, lipids, proteins, and nucleic acids. Get that off there so you can see. Okay, so we're going to talk about just very quickly, let's look at this chart, the differences between organic versus inorganic and organic molecules. Okay, let's start over here with the organic molecules. We know that we have to have carbon. Okay, we'll go back and what you already know about carbon. What is the atomic number? Okay, the atomic number is 6. How many valence electrons does carbon have? Well, with 6 electrons, you're going to have 4 valence electrons in that outer shell. So we talked about that. It's invested way too much. It is going to want to share those electrons. It's not going to want to give those away. So it's always going to create covalent bonds, not ionic bonds. So as you see here, that's a big difference between inorganic and organic molecules. If it contains carbon, it's going to be a covalent bond. If it doesn't, then it's going to probably make an ionic bond, which we learned if it makes an ionic bond, you're going to end up with positive and negative ions. Hopefully you remember the positive ion is called a cation. Negative ion is an anion. Okay, so if you picture this carbon over here, it has 4 in the valence shell, which means it wants 4 more electrons in order to be completely happy. So when it makes this covalent bond, there's going to be lots of other atoms that it can bond with in order to fill its shell. So you're usually thinking that it's quite large. Okay, there's a big molecule. Lots of things can stick to this carbon and hydrogen. Over here, if it's ionic bonding and you're just transferring an electron, it's probably just one transfer to another molecule. Therefore, they're going to be smaller. And then inorganic molecules are associated with non-living things. Organic molecules are then in living organisms, which are the biomolecules. So there's 4 biomolecules. We'll separate those out and talk about each one separately later. But first, we want to talk about the structure of organic molecules. Okay, so if they're organic, like we said, they got to have carbon, hydrogen in them. There's 2 things about them. They have a carbon skeleton and they have a functional group. The carbon skeleton, or the backbone, is referred to as the carbon chain. In the chart over here, it's going to be R. R is going to be standing for the remainder. So the entire remainder of this molecule is the carbon backbone. Whether it's a bunch of C's put together with other C's, how many H's are in there, that's the whole remainder of that entire molecule. Now, I know when you're reading your book, it talked about the backbone of the vertebrae of an animal. Okay, so if you're a vertebrate, okay, you've got a backbone, but what's attached to that backbone makes a totally different organism. So even though you've got a backbone, do you have flippers attached to it? Do you have wings attached to it? Do you have arms attached to it? What are those appendages? So what is attached to the backbone makes a very big difference. Same thing if you look at this molecule. Okay, so the remainder of the whole molecule is the backbone that we're not really seeing. And then what's attached to it can be totally different. That's called the functional group. Specific atoms bonded to the carbon skeleton with characteristic structures and functions. It is this functional group. Look, I started twice. It must be very, very important. These functional groups are what determine the chemical reactivity and polarity of organic molecules. So it's whatever is attached to the backbone is going to go and connect with other molecules. So the chemical reactivity, how it's going to chemically react with others, is all determined by the functional group. Okay, so we want to talk about an isomer. These are organic molecules that have identical molecular formulas, but a different arrangement of atoms. Remember, we talked about structure determines function. Structure always determines function, like we were talking about cells. Okay, look at a sperm cell having a flagella, so it can swim. You've got a red blood cell with, looks like a little donut, so it can hold the oxygen. Okay, so the shape, the structure determines its function. Same with a molecule. So what you're looking at here, this molecule has C3, and then it's got 6 H's, and then it's got 1, 2, 3 O's. So C3H6O3. This one, totally different, but has the same formula. C3, there are 1, 2, 3, 4, 5, 6 H's and 3 O's. So both of these are C3H6O3. But they are totally different functioning because of the arrangement of where these atoms actually are. So structure, again, determines function. Now, we all know glucose. Hopefully, everybody knows the formula for glucose, C6H12O6. Okay? Well, we always talked about that as a molecule, of course, in biology. But it actually has 16 isomers for glucose. So there are 16 different C6H12O6s out there, depending on the arrangement of all of those different atoms. And of course, then they're going to have a different function based on where they are arranged. So you really need to dive in and look at this molecular arrangement and see where those atoms are. Okay, a macromolecule. They're going to be made up of monomers. Okay, so a monomer is a single unit. Use your prefixes. Mono means one. Okay, so think of a monomer as one individual Lego. Okay, so if you put a bunch of Legos together, you put a bunch of monomers together, you're going to get a polymer. Again, the prefix poly means many. So you've got many monomers bound together. So then we just kind of use some language here. It says monomers, the single units, are polymerized, which means joined together to form a polymer. So in the picture over here, polymerization is taking place. They're being polymerized. It's hard not to say being polymerized. You cross-country people have been polymerized. These are polymerized. Polymerized. Okay, so it's a little bit different to say. But this is what those biomolecules are made up of. Biomolecules are big macromolecules that have these monomers making up their big units. Okay, so how do you make up a big polymer? Okay, you're going to have to put monomers together, which we know that's called polymerization, polymerized, being put together. This is a chemical reaction that has to take place. This chemical reaction, if you're putting monomers together, is called a dehydration reaction, or you can also call it a condensation reaction. So if you look at this picture, if you are building or synthesizing a biomolecule, you're putting these together, you're going to push out water in the process. Okay, so as these two monomers right here come together, water is squeezed out. Hence, dehydration reaction. If you're squeezing out water, you're dehydrating something. Or condensation reaction, making water, so they're squeezing it out, forcing the water out. Okay, so when they do that, push two monomers together, squeezes out water, this is a very long polymer now. Okay, so then just the opposite can take place if you're trying to go from a polymer and break it down to the individual monomers. That is called hydrolysis. Okay, so that's just going the opposite direction. Breaking down polymers. Then if you want to you're starting at the bottom. You want to break down this big polymer, you're going to add water. Hence the word hydrolysis, to hydrate, to add water to something. So a hydrolysis reaction puts water in, which now you have your separate monomers back again. So that's degradation of a molecule, degrading it, breaking it down. Okay, so whether you want to synthesize, build up a molecule, that's a dehydration reaction. If you want to degrade it, break it down, that's hydrolysis. Now, easy way to kind of picture this. Take this polymer right here, and let's pretend I've got a couple of you standing in front of the classroom, and you're standing out holding hands. Like back in the day when you played Red Rover. Send somebody. You send somebody right over. Hopefully, everybody played Red Rover. It was a great game. Okay, so you're holding hands, and when someone comes over, they break your bond. Just kind of picture a big fire hose. Okay, if I had a big fire hose and I aimed it at you guys holding your hands, it would break your hands apart. Okay, so if I add water to your holding hands, I can pull something apart. That's kind of how I picture this big polymer. They're holding hands. We got this bond. I add water to it and it breaks them apart. Okay? So this picture just summarizes the biomolecules, and you've got carbohydrates, lipids, proteins, nucleic acids. Those are the four biomolecules. This has subunits. That would be the monomer. So that's the single little building block or Lego piece, and you put it together, you get the full polymer, a very large molecule. So in a carbohydrate, the monomer is a monosaccharide, okay, meaning one. And if you put a bunch of monosaccharides together, eventually you could get a very large molecule called a polysaccharide. There is one in between, the disaccharide, which you put two together. Okay, look at the lipids. Their monomer is glycerol and fatty acids. You put a bunch of those together, you get the entire polymer, which is a fat. Proteins, the monomer is an amino acid. Put a bunch of amino acids together, you get a polypeptide. And remember, poly means many, so that you're thinking a large molecule. Nucleic acids, one little single unit is called a nucleotide. Remember, phosphate sugar base. You put a bunch of phosphate sugar base, phosphate sugar base, phosphate sugar base, you are making a polymer, a large molecule of either DNA or RNA. So all of our big molecules are made up of little tiny units, and those are the monomers. Okay, that's all of, no, it's not all. We got a couple more slides. Last slide, synthesis and degradation. In other words, building up or breaking down biomolecules. Because these are chemical reactions, all chemical reactions require enzymes. Now, just a quick question back from our first chapter, characteristics of life. Which characteristic of life are we referring to when we talk about all the chemical reactions going on in the body? Give you a second. Hopefully you all said metabolism. Okay, so these chemical reactions require enzymes. And an enzyme, which we're going to see later, it's actually a protein, and it speeds up a chemical reaction. It's not consumed in the reaction, and it's not changed, so we can use it over and over again. But Otherwise, it would take way too long for these chemical reactions to take place. So an enzyme is needed for both synthesis and degradation. All right, that's it for chapter 3.1. And then we're going to dive into these biomolecules singly. That's next.